So, single cell sequencing sequences individual cells. So the first question that arises here is, how do you obtain individual cells? Well, there are two systems for doing that. On one side, you have the droplet-based systems. 10x Genomics is the most well-known example of this. On the other side, you have plate-based systems, where you have BD Rhapsody, SmartSeq, ParsBio. So in the droplet-based system, you work with an oil solution, and in that oil solution, there are small water droplets. So it's like an inverted mayonnaise, basically. And in these water droplets, you can inject individual cells. So in this way, you get water droplets that each contain one single cell. In the plate-based systems, you're going to work with microwell plates. So typically, you use 384 well plates, and you distribute your individual cells in such a way that every well will contain one single cell. So that means that in this system, you're going to sequence only a few hundreds of cells, typically 384 cells, because you have 384 wells in the microwell plate. Here in the droplet-based system, you can sequence thousands and thousands of cells. The protocol that is used to obtain the single cells is more harsh in the droplet-based systems, because you literally have to inject them into the water droplets. And as a result, if you want to sequence fragile cells, you might be better off using a plate-based system than using a droplet-based system. So in the droplet-based system, you can sequence a lot more cells, but for fragile cells, the plate-based system will be better. In this course, we will focus on the droplet-based systems because most people generate 10x genomics data, but the pipeline that we show is exactly the same when you start from BD Rhapsody data or even from SmartSeq or ParsBio data. Sequencing for 10x genomics is done on the Illumina sequencers, so the same sequencers that are also used for bulk RNA-seq. Illumina sequencers generate short reads, around 100 base pairs, and what is done is paired end sequencing. That means that you are going to sequence the two ends of your transcript. So you're going to sequence both the 5' and the 3' end of your transcript. Now, on the 5' end, your transcript will typically contain here this sequence that comes from the surface of the bead. So one sequence that you generate during your paired end sequencing will contain the barcode and the UMI sequence, and it will not contain a sequence from the transcript, only the barcode and the UMI. The other sequence that you generate on the other end of the transcript will contain 100 bases of the end of your transcript. So that's what you see here. You generate paired end data, but one end is just your barcode and your UMI. The other end is the actual sequence from the transcript. In SmartSeq data, this is not the case. You will sequence here the full transcript over the entire length, and this makes SmartSeq data much more suited for looking at splice variants than 10x genomics data. If you only sequence the last 100 bases of your transcript, you can't say anything about splicing. If you sequence the full length of the transcript, you can study the splicing.